Jesus said, for judgment I've come into this world. And it is time to begin. I see the sun's out. Nice day today, even though it's cold. So let's have a word of prayer. Let's begin. Our gracious Heavenly Father, we thank you that you give us sight. You let us hear you. You let us see you. You let us obey you. Help us now to obey you in our studies. Thank you. We have the end in sight. Now we pray you would bring us there. Lord, may we not give up. May we not faint. Let us not lose heart as we go to the end of this semester. In Jesus' name I pray. Amen. All righty. Beta-oxidation. We got the fats from the diet into the cell. You know, there's one thing about that I did not mention. So when the fatty acid enters a cell through the fatty acid transporters, and there's several of those, they don't just get stuck in the cell and then they float around until they find where they're going. They're actually bound by an intracellular fatty acid binding protein, an FABP. There are 9 different genes. Got that right? I think it's 9 different genes in the human genome that encode fatty acid binding proteins. 4 of them are actually grouped together on the same part of chromosome 8. That's a tidbit of worthless information. Some of these are expressed in specific tissues. So there's some that are liver specific, adipose tissue specific, heart specific. In cells that do a lot of fatty acid handling, they can account for up to 5% of the protein in the cell. And they're essential for binding these hydrophobic ligands to reduce the detergent-like properties of fatty acids, of at least high concentrations of fatty acids. There's also a group of transcription factors. They're called PPAR. There's an alpha, PPAR alpha, PPAR gamma, and a PPAR beta transcription factor, and these bind fatty acid ligands. And these fatty acid ligands are brought to the PPARs by these fatty acid binding proteins. So these PPAR stands for peroxisome proliferator activated receptors. Peroxisome proliferated activator receptors. They play a very important role in glucose metabolism in general. So these FABPs are quite important when it comes to the various jobs that they do. All right, let's now identify the steps of beta oxidation. Beta oxidation will have an oxidation step, a hydration step, another oxidation step, and a thiolysis step. So first and third steps are oxidations. Second step is a hydration. Fourth step is a thiolysis. Thiolysis. You produce acetyl coenzyme A, and you produce a fatty acid that is 2 carbons shorter than it started. So here is the process at a glance. You start out with an acyl coenzyme A dehydrogenase. It will oxidize the bond between the alpha and the beta carbon. You make a trans, and this is very important, trans Beta-2-enoyl coenzyme A. So you go from the, let's say it's palmitoyl coenzyme A, you make a enoyl, trans-enoyl coenzyme A. Then you add a water across that double bond, and you make a beta-hydroxyacyl. Very important, you make an L-beta-hydroxyacyl, because the next enzyme can't handle the D stereoisomer. It can only handle the L. And the enoyl coenzyme A hydratase can only hydrate trans delta-2-enoyl coenzyme A's. Then you're going to reduce that again, and you'll make a beta-ketoacyl coenzyme A. And then you add a coenzyme A. And the sulfur on the coenzyme A is going to attack this carbonyl carbon right here, and it's going to form a bond. Oh, sorry, no. It's going to attack this carbonyl carbon here, and you'll get an acyl coenzyme A and a myristoyl coenzyme A here, which is 2 carbons shorter than the palmitoyl.
Does the process keep going until?
Yep.
Okay.
Process keeps going. So if you have a 16 carbon fatty acid, then you're going to have to subject that to 7 rounds of beta oxidation.
Okay.
And those 7 rounds, 1, 2, 3, 4, 5, 6, 7, those will give you 8 acetyl coenzyme A's, which will then get oxidized by the Krebs cycle, and their reducing equivalents will get oxidized by the electron transfer chain to produce water from molecular oxygen and ATP. Okay, acyl coenzyme A dehydrogenase has 6 different isozymes that are specific for fatty acids of different lengths. There is a so-called ACAD-M. ACAD-M is the medium chain fatty acid acyl coenzyme A dehydrogenase. Then there's an ACAD-S for the short chain fatty acyl coenzyme A's. And there's some others too as well. These are definitely the main ones. And there's a long chain, which will handle fatty acids from 12 to 18 carbons long. What if they're longer than that? Well, if they're longer than that, they tend to go to the peroxisome first. They get cut down to size, then they get to the mitochondria. More on that later. So the mitochondrion is usually going to handle things within the 20 to the shorter range.
Mhm.
So then you have the medium chain, ACAD, so ACAD-M, and then ACAD-S, which will handle between 4 to 8 carbons long. So there's also a ACAD-VL, which is a very long acyl coenzyme A, acyl-CoA dehydrogenase. So this will produce an oil coenzyme A and a reduced flaving. And that reduced flaving goes straight to complex 2. So this particular step is linked into complex 2, and those electrons go straight into the electron transport chain. And there you are. So the FAD will tend to be enzyme bound and will tend to be associated with complex 2. That gives you the trans delta 2 NOEL coenzyme A. Now, if you eat unripe aki fruit, and this is found in the islands, also the mainland of Central America, it contains this unusual amino acid called hypoglycin A. Hypoglycin A is a suicide inhibitor of acyl coenzyme A dehydrogenase, and this will cause so-called Jamaican vomiting sickness. Jamaican vomiting sickness. So if you're going to eat aki fruit, don't eat unripe aki.
What does mechanism-based inhibitor mean?
Mechanism-based means it's suicide-based because the mechanism takes advantage of the mechanism to make a covalent adduct with the enzyme permanently killing it.
So it's
That's
just a permanent inhibitor?
Yep.
Okay.
Because the mechanism provides, it's a substrate for the enzyme, and the mechanism provides a way that it gets activated and makes a covalent derivative out of the enzyme, which kills it. All right, step 2. Is the NOEL coenzyme hydratase. Hydratase. This just adds a water across that double bond, producing the L-hydroxyacyl coenzyme A. L-beta-hydroxyacyl coenzyme A. And you can see that here. Now we have another dehydrogenation, another oxidation. This is going to reduce NAD. And the reduction of NAD will produce NADH. And so we have another NADH for the electron transport chain, don't we? and that produces a beta-keto-acyl coenzyme A. This is sometimes called a 3-hydroxyacyl-CoA dehydrogenase. It's called that too, because you have carbon number 1, 2, 3. So it gets called beta-hydroxyacyl coenzyme A, and it gets called 3-hydroxyacyl coenzyme A. You'll see both names. And then the final step is catalyzed by acyl coenzyme A acyl transferase. This enzyme is also called thiolase or beta-keto thiolase. Thiolase is definitely the most common name. It's also called acyl-CoA acyl transferase. It's called that too. So acyl coenzyme acyl transferase is commonly called ACAT. ACAT. And there's several different ACATs encoded by our genome. So acetyl coenzyme A is very stable. It's a good leaving group. The sulfur on the end of the coenzyme A is the attacking group. So it creates a new thioester. And so the thiol group cleaves the substrate, so it's a thiolysis. Thiolysis. And that gives you acetyl coenzyme A and a shorter fatty acid to oxidize. And the recipient of all that coenzyme A is a Krebs cycle. And the recipient of all those high energy electrons created by the oxidative reactions of the Krebs cycle is the electron transport chain. And the recipient of all those high energy electrons going to the electron transport chain is molecular oxygen to produce water. As illustrated here by this figure, shamelessly stolen from your book. All right, let's do the math and add this all up. So each oxidation of each acetyl coenzyme A to produce by beta oxidation gives you 3 NADHs, 1 FADH2, 1 GTP, and your PO ratio for each NAD is going to be 2.5 ATP. For each flavine, reduced flavine is 1.5. And then beta oxidation for each production of each acetyl coenzyme A gives you a single reduced NAD and a single reduced flavine. That gives you 4 NADH, 2 reduced flavines, 1 GTP per acetyl coenzyme A generated. Then we apply this to our C16 palmitic acid. So we get 7 cycles. So we get 7 of these, producing 1 NADH, 1 FADH2. We have 8 acetyl coenzyme A's. 8 of those oxidize in the Krebs cycle. 8 times 3, 24. 8 times 1, 8. 1 GTP. 8 times 1, 8. So 8 plus 8 plus 24. That's 40, I believe. I did that right? Then we throw in beta oxidation on top of that, and that gives us 108 ATP per palmitic acid. 108 ATP per palmitic acid. Now we had to burn 2 ATP, at least the equivalent of 2 ATP, for the entry of the ATP into the mitochondria, because of the conversion of the fatty acid into fatty acyl coenzyme A, and then the transport across the membrane via the acyl carnitine acyl coenzyme A transporter. So we subtract those two, that's 106 ATP per molecule. Now let's compare this to glucose. 32 ATP from a 6-carbon glucose molecule. That's a carbon to ATP ratio of 5.3. Palmitic acid, carbon ATP ratio of 6.63. Fat gives you a lot of energy. A lot of energy.
Now, smaller chain fatty acids will have a smaller carbon to ATP ratio, correct?
No, they're the same, because the ratio will be the same. The amount that you make, but the amount per carbon that you make will be the same.
Would still be 6.63?
Yeah. If you start introducing double bonds, that'll lower it, but it still won't lower it to the level of sugar. Now, what happens if you are introduced to unsaturated fatty acids? Particularly unsaturated fatty acids that you get from plants, which tend to have lots of very interesting fatty acids with double bonds in lots of places. So the enzyme NOEL, coenzyme A hydratase, cannot recognize double bonds. They're in the cis conformation. So if you want to add a water across that bond, you have to take that cis conformation, convert it into the trans conformation. Are there enzymes that'll do that? The answer is, but of course. Since just about all unsaturated fatty acids that you get from plants and what have you contain double bonds on the cis conformation, you have to be able to do this reaction. So this is a very important reaction when it comes to the oxidation of unsaturated fatty acids. So here is an important enzyme. It's called NOEL coenzyme A isomerase. NOEL coenzyme A isomerase. This will convert the cis delta 3 NOEL coenzyme A to the trans delta 2 NOEL coenzyme A molecules, and now your beta oxidation continues without a hitch. This enzyme only works on unsaturated fatty acids with double bonds between carbons 3 and 4, between the beta and the gamma carbons. So here, for example, we have oleic acid. We have 2 rounds. And then a 3rd round of beta oxidation. We get 3-acetyl coenzyme A. We now have this puppy here, a cis delta 3 dodecanoyl coenzyme A. You need to convert that to the transform. Fine. NOEL coenzyme A will do exactly that. You convert that now to the trans 2-dodecanoyl coenzyme A. And now you can just continue. You hydrate that, oxidize that, polylase that, and you're fine.
So that that enzyme doesn't just rearrange, I guess, the hydrogen to make it from cis to trans. It also changes the placement of the double bond.
Yeah,
Okay.
yeah, it's a true isomerase in that it will not change the location, but it changes the configuration. And it's a really important enzyme.
It looked to me as if it changed it from the third the third carbon to the second carbon.
Oh yeah,
That's what I
It
meant. So
certainly
it does
does.
change. It changes the position of the double bind.
yeah.
Yeah.
It certainly does. And the configuration then of the bond itself. So it not only makes a cis double bond, makes it trans, it flips from one side to the other side.
Right. Okay.
It's a very, very useful enzyme. And in some cases, you get double bonds that are a right bit tougher to handle. So for example, linoleic acid contains two double bonds that are kind of sort of just across the alley from each other, 9 and 10, and then 12 and 13. So this gives you a configuration where you have double bonds at the beta and the gamma, and then the epsilon and the eta carbons. And this is called a cis delta 3, cis delta 6 configuration. And the enzymes of beta oxidation look at that and go, huh? So that needs to be fixed. That needs to be adjusted before beta oxidation can work on it. And this will require a 1-2 punch with two different enzymes. And so the first of these is an NOEL coenzyme A isomerase. The second is an NOEL coenzyme A reductase. And I'll show you how these work in just a bit. These enzymes often work together. It's not uncommon to see these enzymes forming complexes. And these complexes sometimes are able to work together in this sort of thing, this sort of arrangement. So you then rearrange it after so you rearrange, reduce, and then rearrange again. And so here's your linoleal coenzyme A, and you do 3 rounds of beta oxidation. Now you get a cis3, cis6. The enzymes of beta oxidation look at that and say, yeah, I don't think so. So the first thing that happens is the NOEL coenzyme A isomerase comes in and flips this bond here to here. So cis converts it from cis to trans. Okay, good. Now I can have one more round of beta oxidation. Now I have a trans to cis 4, egads. That's not something that beta oxidation can work with either, because this hydratase cannot work if there's another double bond across the alley. So what you're going to do is you're going to get rid of one of these double bonds. You're going to get rid of this one. That's going to flip this double bond to over here between 3 and 4. So that's the dienoyl coenzyme A reductase. That flips it to here, and then the enoyl coenzyme A reductase. Even though this is a trans, this is in the wrong place. So Irma, that kind of gets back to your question. This enzyme here doesn't flip it from cis to trans. It just flips the location of the bond here. Now, this is a substrate for beta oxidation. The hydratase will handle this, and then you go from there. So it's this 1-2 punch of these enzymes that essentially take these cis3, cis6 enzymes and make it possible for beta oxidation to work with them. Now that's all fine and good, but what happens if you have an odd numbered fatty acid? Are all fatty acids even numbered? Oh, if only that were the case. No, you have plenty of odd numbered fatty acids out there. So you're going to oxidize it to propionyl coenzyme A. What happens if you have propionyl coenzyme A? You have a pathway for dealing with propionyl coenzyme A and for dealing with odd numbered fatty acids. So the first of these will be the enzyme propionyl coenzyme A carboxylase. So this enzyme will use ATP, biotin, and CO2. So it's an ABC enzyme, an ABC enzyme. And that's going to make D-methylmalonyl coenzyme A. Just like carboxylases that use biotin, this is going to require the activation of the biotin. That requires the signature of an ATP. So from this, you make D-methylmalonyl coenzyme A, then you rearrange it to L-methylmalonyl coenzyme A. Because the next enzyme, a mutase, can't work with the D-methylmalonyl coenzyme A. It must have L-methylmalonyl coenzyme A. So to rearrange that, you're going to use an enzyme called a methylanyl coenzyme A epimerase. Epimerase. Then you have a mutase, which is a B12 containing enzyme, and it converts the L-methylmalonyl into succinyl coenzyme A, and succinyl coenzyme A is ready-made for the Krebs cycle. So even if you have an unnumbered fatty acid, it still goes to the Krebs cycle. So here we have our propionyl coenzyme A. Propionyl coenzyme A carboxylase is a heterododecameric enzyme. It has an alpha-6, beta-6 composition, and the two different subunits are encoded by the PCCA and PCCB genes. This has the same mechanism that we saw with the pyruvate carboxylase enzyme. Same mechanism. Then comes the rearrangement, epimerization. So you can see this group here and this group here are going to swap. That's your epimerase. Your epimerase. Then you have the mutase, the M-MUT protein, methylmalonyl coenzyme A mutase, that uses a vitamin B12 cofactor. And it converts this now to, it's going to take these two and swap over. And now you make methyl, from methylmalonyl coenzyme A, L-methylmalonyl coenzyme A, you make succinyl coenzyme A. And the B12, you can see the mechanism here. You make a free radical. That free radical will abstract the hydrogen. You then make the methyl deoxyadenosine. Then you have a radical rearrangement so that this radical gets shifted over here. Then this is going to give the hydrogen back to the product here. And then this will reattach to the cobalt, and you've regenerated your cofactor. So we see a trend when it comes to the enzymes of beta oxidation. If we look at bacteria, we can see that the gram-positive bacterial genes encode proteins that have a loose affinity for each other. And they pass their products to each other largely via diffusion. This is also the case for the mitochondrial short-chain fatty acid system. In gram-negative bacteria, we see that enzyme 2 and 3 are encoded by a multifunctional gene that encodes a multifunctional polypeptide. In mitochondria, for the long chain, very long chain specific system, we see something here that looks a little bit like what we see with gram-negative bacteria, don't we? And then with plants, we see something really interesting where this enzyme activity is merged to several other proteins as well. So we can see sometimes we have multifunctional polypeptides encoded by very long genes that essentially make sure that the substrates are passed directly to each other in order to make sure this process occurs very quickly indeed. What about the oxidation of phytanic acids? Phytanic acids are the result of isoprene chains. If you ate, let's say, a carrot for lunch today, or if you had any colored foods, if you had lettuce or spinach or bell peppers or Brussels sprouts or any sort of plant material, you ate some chlorophyll. Chlorophyll has a phytan chain on it. You think your body's going to clip that off and get rid of it? No. That's an important carbon and energy source. Same thing with carotenoids, same thing with anthocyanins, so on and so forth. These are also found in dairy products. They're also found in the tissues of ruminants. If you've had any steak or a burger lately, you've probably eaten these sort of phytanic acid derivatives. So these cannot act as a substrate for any of the enzymes of the mitochondrial beta oxidation pathway. Instead, these are a substrate for the alpha oxidation pathway, and that occurs in peroxisomes. So this requires a special set of enzymes. So you have a phytanoyl coenzyme A synthetase that makes a phytanoyl coenzyme A. This reaction occurs in a cytosol, and then the rest of the reactions occur in the peroxisome. So you have a hydroxylase that will make an alpha hydroxy phytanoyl coenzyme A. You can see why it's called alpha oxidation. Then you have a lyase, alpha hydroxy phytanoyl coenzyme A lyase, which will make formyl coenzyme A, which is burned to CO2 and formic acid. Then you have a aldehyde, okay, and you convert that aldehyde through oxidation to a carboxylic acid, and you make persenic acid. That is now a substrate for beta oxidation. That will produce propionyl coenzyme A, and propionyl coenzyme A will actually be shipped to the mitochondrion for further oxidation. And the medium and the short chain fatty acid will be esterified to carnitine and will be transported to the mitochondrion for further oxidation. Questions? Okay, no questions. Then let's soldier forth. So let's talk about the regulation of fatty acid oxidation. So fatty acyl coenzyme A molecules that are bound to the fatty acyl binding proteins, they can be either moved into the mitochondrion for beta oxidation, or they can be converted into fats for storage or be used to make phospholipids. Triacylglyceride and phospholipid synthesis occurs in the ER or the Golgi if you want to make a glycolipid or a sphingolipid. Those are the only two fates of fatty acyl coenzyme A's. How does the cell decide between one or the other? It depends upon the rate limiting step, that is the entry of these molecules into the mitochondrial matrix. Once you get the fatty acyl coenzyme A into the mitochondrion, they're committed to beta oxidation. There's just no two ways about it. And that makes sense given that the entry is the limiting step. So an excess of fatty acyl coenzyme A molecules will push some of them into the synthetic pathway. excess glucose will be used for fatty acid synthesis, triacylglyceride synthesis, which will be used to store energy. Now, the first molecule made during the process of fatty acid synthesis is the carboxylation of acetyl coenzyme A to make malonyl coenzyme A. Malonyl coenzyme A is a potent inhibitor of carnitine acyltransferase 1. Carnitine acyltransferase 1, you will, I hope, remember, is the isozyme of carnitine acyltransferase that faces the outer mitochondrial, embedded in the outer mitochondrial membrane and faces the cytosol. So malonyl coenzyme A will shut down the transfer of fatty acyl coenzyme A molecules into the mitochondrial matrix. Acetyl coenzyme A carboxylase is a very heavily regulated enzyme. So insulin is going to activate a phosphatase that dephosphorylates it and activates it and causes it to make more malonyl coenzyme A molecules, which will inhibit beta oxidation. So high blood glucose will tend to promote insulin secretion, which promotes acetyl coenzyme A carboxylase activation, because it promotes malonyl coenzyme A synthesis, which inhibits the transport of fatty acyl coenzyme A molecules into the mitochondrial matrix, and therefore promotes fat acid synthesis and fat synthesis, triacylglycerol synthesis, and fat storage. Glucagon, on the other hand, which is made when blood glucose levels are low, promotes protein kinase A activity, which phosphorylates acetyl coenzyme A carboxylase that inactivates it. So now, glucagon inactivates acetyl coenzyme A carboxylase, which decreases the concentration of malonyl coenzyme A, which therefore de-inhibits carnitine acyltransferase 1, thereby promoting the transport of fatty acyl coenzyme A molecules into the mitochondrial matrix and therefore promoting beta oxidation. So when glucose levels are low, you burn fat. What about the other enzymes of beta oxidation? Thiolase. Thiolase. So thiolase is inhibited, first of all, by excess product, acetyl coenzyme A, and the enzyme before it, the beta-hydroxyacyl coenzyme A, the L-beta-hydroxyacyl coenzyme A, the hydrogenase, is inhibited by high NADH-NAD ratios. So it is also end product inhibited. So these two enzymes show end product inhibition. The oxidation of fatty acids occurs in mitochondria, at least in animals. However, the peroxisome also plays a rather important role in fatty acid oxidation. So peroxisomes will tend to handle very long chain fatty acids. By very long chain, I mean C18 to C26. Those will be preferentially oxidized in the peroxisome. Peroxisomal fatty acid oxidation does not proceed to carbon dioxide and water, like in the mitochondrion. Instead, the chain shortened fatty acids will be made into chain short fatty acyl coenzyme A's, or their carnitine esters will be transported out of the peroxisome, which will then be taken up by the mitochondrion, and their beta oxidation will be completed to carbon dioxide and water. So the peroxisome will specialize in the oxidation of cristanes, like we saw. Alpha oxidation occurs in the peroxisome. Several long-chain polyunsaturated fatty acids are first oxidized in the peroxisome. And then many of your prostaglandins and nucleotrienes will be first oxidized in the peroxisome. And so will bile acid intermediates. Now in plants, virtually all fatty acids are oxidized in the peroxisomes. And they work with glyoxysomes to take fats that are stored in, say, seeds and convert them into sugars with the help of the glyoxylate cycle. In animals, it's a different story. So in mitochondria, the acyl coenzyme A will produce a reduced flavine. And that reduced flavine is going to generate electrons for the electron transport chain. That's not the case in peroxisomes. So in peroxisomes, you have a different enzyme. You have this enzyme. They're acyl coenzyme A oxidases. So alcox-1, alcox-2 are the most common ones. They essentially, instead of transferring those electrons to electron transport chain, the flavine gives them to molecular oxygen, which produces hydrogen peroxide. And then hydrogen peroxide is converted by catalase into molecular oxygen and water. This is the reason why peroxisomes have so much catalase, because they make a lot of hydrogen peroxide. The other steps of beta oxidation in peroxisomes are the same as in mitochondria, except that the NOEL coenzyme A hydratase and the beta hydroxyacyl coenzyme A dehydrogenase are both catalyzed by a bifunctional protein. So the protein in the peroxisome is bifunctional. It does both of those functions. And then you have a peroxisomal thiolase that tends to be specific to the peroxisome. It's called ACAA1. In the peroxisomal membrane, you then have a transporter. That transporter is called carnitine phosphoryl transferase, or CROT, and that will take the medium chain acyl coenzyme A's, transport them out of the mitochondrion into the cytosol where they'll go to the mitochondria. So they go to the cytosol to the mitochondria. Plants, of course, will use glyoxysomes, predominantly in germinating seeds, peroxisomes in leaves. Those are the organelles that will oxidize your fatty acids in plants. Acetocoenzyme A, made by peroxisomes and glyoxysomes, will be used by the glyoxyl pathway for biosynthetic purposes. And so plants will use this acetocoenzyme A to make amino acid, carbohydrates via gluconeogenesis, fats via fatty acid synthesis, nucleotides, lots of things made from the oxidation of stored fats. Omega oxidation is also called microsomal oxidation. This oxidation essentially will begin with the production of a carboxyl group at the far end of the fatty acid, thus producing a so-called omega carbon oxidation. So this reaction, as you can see, occurs in the ER. Drug detoxification tends to occur using this type of biochemistry, among other types of reactions too. This is one of them. So you're going to use so-called mixed function oxidases. Mixed function oxidases. So these are cytochrome P450s, in particular cytochrome P450A4s and AF family members. We'll add an alcohol to the far carbon of the fatty acid. These are fatty acids that are between C10 to C26. They can be branched, they can be derivatives. These mixed function oxidases are sometimes called monooxygenases. And they use this general reaction here. And they use several different co-substrates. The bulk of them use ADPH. Others use flavines or NAD. There are a couple that even use alpha-ketoglutarate, but they're in the minority. Most use NADPH. And the cytochrome P450s get their name because they absorb light at 450 nanometers when you make a carbon monoxide complex out of them. And this enzyme complex is localized to smooth ER. Smooth ER. So you have your aliphatic compound. You make alcohol out of it. You're going to use molecular oxygen as a source of electrons. You make the alcohol. You reduce your cytochrome P450. You have some sort of cytochrome P450 reductase, which will reoxidize it, and then you'll use that, as you can see, to give those electrons to NADPH or something like that, or what have you, or perhaps to FAD or NAD or something like that. So these reactions are pretty heavily used in liver cells. They'll be used to degrade several different fatty acids that are used in signaling in some cases. After you make the alcohol, you then subject it to alcohol dehydrogenase, which converts the alcohol to an aldehyde. And then you use an aldehyde dehydrogenase that will convert it to the dicarboxylic acid. And then you have a dicarboxylic acid, which can be degraded via beta-oxidation II. And this essentially shows you how this works. And we're out of time. Okay, we are almost done. So we'll look at ketosis and the synthesis of ketone bodies. This occurs during starvation. It's a very heavily rated process, as you might guess. So we will pick this up on Monday. It most certainly won't last all of Monday. And I hope you guys all have a great weekend. And I will see you.
I have a question.
Sure.
So I emailed Kathy about my quiz 8, and she was wondering if she can just email it to me or if she wanted me to do it over Zoom.
Oh, she can email it to you.
Okay, I'll let her know then. Thank you so much. Have a great weekend.
Dr. Radovich, is that what is made, are ketone bodies made during the keto diet that people are doing?
Yeah, that's the logic behind it, is to not give yourself glucose. You eat mainly protein and that sort of thing. You don't eat any sugar.
Right.
So it forces your body to live off ketones. And by living ketones, you will potentially burn more fat. Because burning fat during starvation is used to make ketones.
Is that a good idea for your brain anyway?
The research on the keto diet kind of goes back and forth. Most of the studies that were done were small studies. There was a bigger study that was done at Mayo Clinic, and the bigger studies showed that people on the keto diet didn't lose any more weight than those on a non-keto diet. So also, people on the keto diet, tended to have less, they had subpar brain activity, and they also had subpar activity in other organs too. So the recommendation was from this research group, if you use the keto diet, use it short term, don't use it long term.
I know at least one person that's doing it because they have cancer. And so the idea is that if you starve cancer cells from glucose, does that have any bearing on it?
Well, cancer cells are subject to the so-called Warburg effect, where they take up lots of glucose. Because their mitochondria tend to be shot, they'll tend to make most of their energy via glycolysis. They'll make large amounts of lactate, and by potentially starving cells for glucose, you deprive cancer cells of a major source of their metabolism. The problem is that cancers are very good at adapting. And so while you might get cancer cells to, at least in some parts of tumors, particularly in the interior of tumors, to maybe start to give up the ghost, other cells are going to adapt. So starving cancer cells might work if you're hitting them really hard with chemotherapy, but cancers are very good at adapting. So there's not really any good clinical evidence, albeit I have to admit it hasn't been studied all that much, that a keto diet is going to give you more anti-cancer bang for your buck than a more balanced diet. Now, there are some oncologists I've talked to who swear that if you load up on antioxidants while you're taking chemotherapy, the cancer cells actually use the antioxidants to protect themselves from the chemotherapy. So they will often tell their cancer patients, don't load up on your supplements, your vitamin E or whatever, because the cancer cells then will tend to fare better from the chemotherapy.
Interesting.
Another oncologist I talked to said that he actually recommends for certain cancers, because certain cancers that he's seen that are very glucose hungry. So he said, I start patients on a keto diet, I think something like 2 weeks before chemo. Then when they go into chemo, the cancer cells are, as he put it, the cancer cells are pissed off. So when you hit them with chemo, they die much faster. That's his experience. I said, have you studied that? Have you published that? He said, no. That's just my protocol. It works, so we go with it. I said, for goodness sakes, study this. And he said, I can't get any money to study it. So some people say, at least for some cancers, it works.
But not long term.
Well, this is for this guy. This guy specialized in leukemias. So he said for leukemias, it worked.
Okay.
For solid tumors, solid tumors are a different story. Solid tumors are much more heterogeneous, and solid tumors adapt much, much faster than do leukemias, lymphomas, and stuff like that. So the two oncologists talked to, one of them specialized in breast cancer, the other in stomach cancers. The stomach cancer guy said, oh, no, don't do a keto diet. Your cancer cells, your stomach cancer cells will feast on the ketone bodies, and your patient will be under the table with fatigue. The breast cancer one said, some patients it helps, others it doesn't. I said, well, can you tell from their labs who will be benefited from it and who won't? He said no. She said no. Oh, probably depends on the cancer.
Okay.
So
Just curious.
yeah, 6 to 1, half dozen of the other.
Yeah. Okay. All right.
That needs to be studied more. There's just not a lot of money to study that,
Yeah. Well,
unfortunately. So
there hasn't been for a while, unfortunately.
yeah, no, that's a good point. So
Okay. Thank you.
I have a quick question too, Dr. Baradovich.
sure.
For the take-home final, is it going to be same rules apply for you can like use your book? And,
Yeah, it's open book but not open neighbor.
Okay, sweet. Thank you. I was just making sure.
Yeah,
Thank you. Have a good weekend.
you too.
And that will be available Monday, right?
Sure. Yeah.
Sometime.
I had a Bible quiz this weekend.
Yeah, I know
I'll
about
work.
that.
I'll work Saturday night and I'll work into Sunday. My wife chews me out mightily when I work on Sunday, but it's finals week. I don't do it every Sunday, but she'll just have to understand. My wife is from that Dutch Reformed tradition where you don't work on Sunday. So I'm just going to have to say, sorry, hon, got to be done. So I'm going to try very hard to get it done by this weekend. So
Well, thank you.
Thank
I
you.
appreciate it.
sure.
I have a couple of questions about the homework from the lab manual.
All right.
So the first one is from the second assignment, the last
Just
part of it.
back off your microphone. You're distorting a bunch.
Is this any better?
That's better. Here, let me turn my speaker down. Maybe that'll help. Yeah, that.
Okay, is this better?
That's much better. Yeah. Yeah, you were distorting a lot for some reason. Go ahead.
Okay, so the first question I have is on number 11 for assignment 2, what in particular you were looking for on the last question
I've got to
for
look at the
numbers
question.
11 and 12.
I got to look at it. I don't know off the top of my head what question that is.
Read it, Jess.
Yeah, I can read it for you.
Go ahead and read it. About the mouse genome and all that?
Yeah.
Okay, you should be able to see then from the search, you're going to get a page full of matches, but a lot of them are going to be in the same gene. So if you click on that
Okay.
gene, you should be able to tell from when you click on that gene, you should be able to see how many tissues it's expressed in. And that'll give you information from the mouse genome database. And that should maybe tell you something about where that gene is expressed. What can it tell you about weight loss, about weight gain, about appetite control? Did the database, did it give you more than just sequenced information? What else did it give you? There's actually a treasure trove of information there if you dig for it.
Okay. And then for assignment 3, the last one, we could, number 5, we could not figure out how to get it to work in the link.
The
Like we
comparing
clicked
the
on
two,
the top link and then we couldn't figure. It
Right,
says retrieve the
you
2
get the
protein
two-
sequences.
right, you get the two,
So number 3 from assignment 3 is the one that talks about getting, like, aligning the 2 with the blast, but number 5 from that same assignment talks about something else. Yeah.
all right, so you get this protein sequence. So there's that sequence. So just take that and take it, put it in a Word document and copy and paste it. And then go to the other one. Copy and paste it. Okay, then you go to and you should be able to put the 2 in. And you have to use the FASTA format where you put the little bracket thing and what have you. And because using protein, you have to use the blast B is already clicked here, and then you click on blast. And that should give you a comparison, essentially, of those two. You should be able to get a readout of the comparison of those 2 sequences.
Okay, that makes sense. I just didn't know what part from the first website we were copying over into the second one.
Right, no, so you get the one and then you get the other, so you, because Zoom is at the top, it won't let me do it. You sequence here, click on that, you should get a sequence comparison out of that. And then it asks you,
Okay.
where are the similarities, where are the gaps. Some people got really disturbed that there were gaps in the sequence. Yeah, there are going to be gaps in the sequence. So then you look for this sequence, and you're actually, these are cyclooxygenases that are involved in inflammation. This strategy was actually used to design drugs like celecoxib and rofecoxib and valdecoxib and stuff like that. It was this type of comparison that led to that.
Okay. That makes sense. And then my last question is the very last question in assignment 4. I got halfway through the question, and then it asks me to click on a link that's talking about the ZZ zinc finger, and I thought I had found it, but when I click on it, it doesn't, when I click on it, there's no link to like the rest of the question. Like there's no show button. So it doesn't tell me what protein I've been looking at.
Okay, so all right, there's your domains, and here you are down here. They changed this. You don't have to click show. It already shows it. There's your domains right there.
Oh, okay.
You just, there's this, there's all that information about this. You can click on that if you want to learn more. There's these different domains here and blah, 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 what have you. There's your zinc finger right there. Zinc finger binding site right there. That's it right there.
Okay, so that says what protein we've been looking at.
Yeah.
Right there. Okay. Sounds good. We just couldn't figure out, I just couldn't figure out the
Yeah,
next
they
page
used
to go
to,
to.
they used to have this. Then you had to click on show for this. Now they made it so that
Okay.
everything's on one page. And they did that just in November.
Okay, sounds good. Thank you.
Could I ask a question on assignment 3, part 2?
Yeah.
I tried to figure out if, so I think I found the sequence of the protein and the ORF13 sequence. However, when I look to see if there's a match, there seems wherever I think, oh, this might be it, but you know that because it starts with similar bases, but then one of them changes. So is it? Are we supposed to just? Could there be gaps in?
Yeah, there could be gaps. There
Okay,
should be
or
an
other insertions.
right. There should be an E value next to it, and that E value, the smaller the E value, the better the match.
Okay.
So you pick the one with the smallest E value.
Let me see. So the E
Okay.
value will appear with the ORF?
It should, well, no, it's, so this asks you to, you click on ORF13, and you take that deduced sequence, and you ask, did the translation of this particular open rating frame, did it match the sequence of the protein that was listed by the blast up here. If there are gaps, that's fine.
So
But then
I
you
would have
do
to,
well enough for it to identify it.
so I click on ORF 13 and it gives me display ORF as either a protein sequence or a nucleotide sequence. So I copied that, but then how do I compare it then to the blast, to the first sequence?
So you take this and you do this business here. That accession number, essentially, that you have from that accession, because this is genome. This is genome. Then you take that particular transcript, you then go to the ORF finder and you ask which ORF, essentially, does the ORF match the protein sequence listed by the blast that you got up here. This will give you a transcript, but that should be a protein that's assigned to that. So when you, oh gosh, I did this yesterday, but it takes a while.
So if I hover over ORF 13, it gives a protein sequence there.
Yeah.
But
So this assignment here in 1, you should have gotten an accession number, some sort of gene that should given you some sort of protein sequence.
right.
Then you put that in here and you're asking it to translate the protein based on different open reading frames. Now, ask the question, did this sequence, protein sequence you got here, does it match the one you got here? That's what I'm asking. And you said there were gaps in it and what have you. If there are a few gaps, that's fine.
Okay. I think I got it. I'll work on it some more. If not, then I will ask on Monday.
Okay.
Thanks.